Okay, hi everyone. Hopefully people can hear me on Zoom as well. So I'm delighted to introduce today's speaker, Dr. Saima from Mount Sinai. He's an assistant professor of genetics and genomic sciences, a member of the Icahn Genomics Institute, and a member of the Skin Biology and Diseases Resource-Based Center at Mount Sinai. He's also an affiliate member at the New York Genome Center. And so Sai earned his bachelor's in chemistry from the Chu Ko-Chen Honors College at Icahn University, and earned his PhD in biomedical engineering from Virginia Tech and the Wake Forest University. He did his postdoc at the Broad, working with Aviv Regev and Jason Van Rostro at the Broad and the Harvard Department of Stem Cell and Regenerative Biology. His lab that I learned started just last year works primarily on innovative experimental and computational tools to investigate epigenetic mechanisms of gene regulation in single cells across dynamic cell fate transitions. And his research combines molecular biology, microfluidics, imaging, engineering, bioinformatics to elucidate the regulatory diversity of single cells within healthy and diseased tissues, including differentiation, hematopoiesis, autoimmune diseases, and tumor progression. And excited to hear more about your work.
Thank you. Thank you so much for the introduction. Just give me 1 second. Hello. Okay, thank you. Thank you so much for having me here today. So today I'm going to talk a little bit about our recent progress in trying to understand the cell fate decision using our epigenomic approaches. So let me start with a very brief introduction of my lab. So as Elm said, our lab started roughly half a year ago at Mount Sinai. So my lab has 3 different components, I would envision. So mostly we are developing sequencing-based technology for either single cell level or in the tissue level. And then we also trying to develop its associated computational approaches, trying to understand the new data that we're generating. We also apply all these different tools into different biological contexts, including tumor progression, including different developmental contexts. So today we're mostly going to cover the technologies and also the computational part, maybe touch base on a little bit on the mechanisms. So as I said, our lab mostly love to focus on the cell fate decision. And when we talk about cell fate, the first thing that came into my mind is this very elegant Waddington landscape model. We know when a marble rolling down the landscape, similar to a stem cell differentiate to different populations, it needs to make a series of decisions, right? So the stem cells can differentiate to progenitors and then further differentiate to diverse downstream populations. But the one thing I want to highlight is that this cell fate decision is really not predetermined. It could be changed by many ways. For example, if we look at this normal hair follicle differentiation, it seems to be a very simple process, but actually the growth and the loss of hair actually go through multiple stages, right? The telogen is a resting phase and the anagen is a rapid growing phase, where the cell growing very rapidly and going downwards, and the catagen is a regressing phase. So you can imagine all the cells here must be sharing very similar genetic information, but they also have very dramatic identity shifts. So this process must be driven by non-genetic factors, right? So it could be epigenetic regulation, could be cell signaling, could be cell spatial location, or it could be some external perturbations such as stress. And we have shown that the hair follicle's fate could be changed by stress. On one hand, we found that long-term stress could actually result in the hair loss. On the other hand, acute stress could trigger gray hair. And you can see President Obama really shows a good example here. So after being a very stressful president for a few years, his hair totally turned gray. So I won't walk you through all the exact mechanisms here, but this really highlights the cell fate could be changed. And apparently there are different mechanisms involved in all this cell fate change. And the one I really care about is the epigenetics. So what is epigenetics? Epigenetics refers to the change without really changing the genomic sequence. It's including different modalities, including DNA methylation, his modifications, and a few others. And today we're mostly going to focus on the chromatic accessibility. So we know we roughly have 3 billion base of DNA in each individual cells. And for most part, the genome is pretty compact. It's called closed chromatin or inaccessible chromatin. But there is a small portion of the genome is actually pretty open. It's called open chromatin or accessible chromatin. And this chromatin would allow transcription factor to bind and then induce the binding of polymerase and then induce the expression of the nearby gene. So largely, we would expect a strong correlation between the chromatin openness and the TF binding and then the expression of a gene. But the exact mechanisms is still largely unknown. And for most part, it's because there's some technological bottleneck. So one simple question we may ask is, how do we even measure this epigenome? So probably the most popular way to measure epigenomes nowadays is using a TAC-seq. It's a method developed by one of my postdoc mentor, Jason, when he was the graduate student at Stanford. Probably many of you are already familiar with what TAC-seq. I'll only highlight this process. Basically, you're using a very special enzyme called TIM5, which is pre-assembled with some DNA oligo. And then once you diffuse this TIM5 transposon into the nuclei, it will cut into the genome and simultaneously ligate the DNA onto the open chromatin. And after that, we can simply purify the DNA and then use that for library preparation. Once we align the DNA library into the genome, we do expect some regions have more reads and some regions have less. And these peaks indicating where the open chromatin is in the genome and the height of the peak indicating how open it is. And usually a TAC-seq is performed in bulk, but it's missing a lot of opportunities to further understand the cellular heterogeneity. And later on, when single-cell TAC-seq method came out, it allows to look into the heterogeneity and also trying to look into the difference between different populations along the differentiation trajectory. For example, in this case, we're looking at the hematopoietic differentiation. We can see each dot here is a single cell. We can follow the trajectory of all the different lineages here. But although single-cell TAC-seq is really attractive, but the IC is still limited by its throughput and also the cost of the IC. If we look back of the history of all the single-cell methods so far, we can see like at the very beginning, we are looking at each individual cell each time. And then later on, when droplet-based methods such as DropSeq and also 10x commercial solution came out, as well as split pool-based barcoding approach came out, we are dramatically improved our capacity for sequencing. Now, roughly, we are looking at sequencing thousands of cells in one single IC. And roughly, it costs about 30 cents to prepare each individual single cell library. It seems to be reasonable, but if we think about how many cells are there in a single tissue, we probably have millions or even billions of cells in one single tissue. So 5,000 cells is really, really not enough. So trying to overcome this bottleneck, several groups, including us, have been trying to improve the scalability and also the sensitivity for single-cell TAC-seq profiling. As one example, we collaborated with Tyler Jack's group at MIT with Lindsay as a postdoc in Tyler's lab. So we're trying to improve the split pool-based single-cell TAC approach and then compare and then look into the trajectory of the tumor progression in the lung. And then we can reconstruct a very continuous chromatin landscape from the normal cells to the metastatic status and then to the eventual metastatic cells. But I want to say that although we are able to reconstruct this continuum landscape, but it has been very challenging in terms of the cell number. In this study back then, it was one of the largest scale study, but still we're only looking at 10,000 cells and looking at 30 different samples. After a few years, by nowadays standard, it's already become like a little bit outdated, but it was a large study back then. And also, we are only able to profile the chromatin landscape only without the gene expression level. And for many of you, you would realize without using the gene expression information, it's going to be very, very hard to interpret what the cell type is and also think about what's the functional result of this method. So to overcome all this bottleneck, I started to develop our own technology. So in the first part of the talk, I'm going to describe how we trying to link the chromatin readout with the gene expression level. So this method, we call it share-seq. The idea is that we want to measure both the chromatin and the RNA from the same single cell in a very, very high throughput manner. That means we want to profile hundreds of thousands individual single cells in one experiment. So in this protocol, we first fix a bunch of cells and then transpose them with TM5 to label all the open chromatin. And then we reverse transcribe the RNA into cDNA with a biotin labeled primer, so that all the cDNA would have a biotin on it. And after that, we split all the cells into 96 well plates, and each well already have a well-specific DNA barcode that can hybridize to both the chromatin and also the RNA, the cDNA. So that means after first round of hybridization, now we would have 96 groups of cells, and each group would have a unique DNA barcode on it. And after that, we put all the cells back together, become one pool, and then split them into second 96 well plate or second round of barcoding. And we repeat this for 3 times in total. That means in total, we would have 96 to the third barcode combinations. And if we took a small subset of cells out of this huge barcoding place, then by chance, we would expect most of the cells would be uniquely barcoded by one barcoding combination. This is how we achieve the single cell resolution without really using any special equipment. And after we took the subset of cells, we can lysis them and then use strip editing beads to pull out the RNA library and then use a supernatant to prepare the DNA library. So because we are using this huge barcoding space, so roughly we can profile about 2 million cells in one single experiment, which is much higher scale compared to previous approach. And the data quality is also amazing, I would say. So if we compare our method, which is this study here, compared to a few competing technologies came out at a very similar time, you would see for both the RNA-seq and also tag-seq, we're getting way more unique information per cell compared to them. Another thing I want to highlight is this method is very affordable and also doesn't really require any special equipment. Depends on how you calculate the cost. It's roughly cost about 0.1 cent to prepare each single cell library, and roughly cost about $400 if you prepare about 100,000 cells. So it's very, very cost effective. To demonstrate the performance of the assay, I did a species mixing experiment. Basically means I put equal number of human cells and mouse cells together and then perform share-seq on it. So we do expect for most part, the genome of all these single cells would align to either human or mouse. And this is exactly what you see here. And out of these 2,000 cells that we sequenced, we identified one single cell doublet and one doublet, now single cells. So the barcoding collision rate is 1 out of 2,000, so it's actually extremely low. So to further demonstrate the assay, we started to applying share-seq in a primary tissue. So we thought about a few different cases, including mouse brain, lung, and skin, and eventually we chose skin as our demo system by collaborating with Yachi's group at Harvard and being as a postdoc in Yachi's group. So skin is a relatively simple tissue, I would say, but still there are more than 20 different cell types in there. I won't ask you to remember all these different populations, but there are a few populations I want you to focus on. So here at the bottom, this is the TAC population, which is known as the progenitor populations. And it's known this one can give rise to a few different lineages, including the IRS here and also the hair shaft. So these are the populations that we're going to focus later on in this talk. So remember in share-seq, we have both modalities. We have the RNA, we have the chromatin. So if we only look at the RNA portion of the data, we can, of course, do dimension reduction. We can cluster the data. And you can see from this U map, we identified more than 20 different populations within the mouse skin. And we can also just look at the chromatin part of the data and also do dimension reduction. We can also just transfer the color from the RNA side to the chromatin side. And already you can, even by your eye, you can already appreciate all the colors still aggregate together, right? That means in ballpark, the cell type definition between the two modalities are very, very similar. And now, because we have both measurements, we can also do regular RNA-seq analysis, like finding marker genes for each population, or we can do regular single-cell tag-seq measurement, trying to look at the marker TF from a tag-seq data for each population. We can also aggregate all the cells together per population to look at its landscape across different populations. You can see the chromatin landscape is also very cell type specific. So now we really have the perfect data to ask, is there any difference between these two definitions, between these two modalities in terms of their cell type definition, right? So to answer this question, I clustered the data using either their RNA profile, which is the row here, and also only on the chromatin data, which is each column, and then ask, what is the overlap between these two definitions? You see a very, very strong diagonal signal here, right? That means, again, in ballpark, the cell type are very similar between these two modalities, but if you look carefully, you'll see some fine differences. For example, at the bottom here, for each of these dermal populations, they seems to have quite distinct RNA clusters, but their chromatin landscape actually look quite similar to each other. And in another case, we know skin is one of the most proliferating tissue. So if we look at the cell cycle gene expression, which is a hallmark for cell proliferation, we can see there are two different populations get highlighted. On the top here is the sublet of the basal cells. And if we track down this sublet, it's a very distinct cluster in the RNA map. But if we track them down in the chromatin landscape, it becomes not one cluster anymore. It's quite spread out, right? That means the chromatin is actually very bad at reflecting the cell cycle information. To me, that means the gene expression level change in the RNA is relatively fast, but the chromatin is relatively slow. The chromatin is having a hard time to actually catch the very dynamic change in the cell cycle. And after we apply shared-seq in skin, we're also trying to apply shared-seq in a bunch of different primary tissues and different contexts, including tissues like airway, brain, colon, lung, T cell, different organoid, as well as trying to apply a huge pool of cancer lines and also in the context of wound healing. So I would probably say like shared-seq is really broadly applicable to many, many tissues at this point. But of course, we don't want to just profile tissues. We want to ask in some real biological questions. So in the field of gene regulation, the key question that we always want to ask is that what are the key genes out of more than 20,000 genes in our genome are important in a given biological context? And then more importantly, what are the regulators actually regulating these key genes? So now with this paired data, we can really ask this question. So to answer this, I developed a computational approach. I call it the cis-regulation approach. So the idea is that for each gene in the genome, I'll try to define a window around this gene, and then simply try to calculate the correlation between the gene expression level and also the accessibility of the nearby peaks. And when we calculate this correlation, of course, there will be a lot of bias from the assay. So we perform the permutation test, try to remove all the bias, including the GC bias and also the accessibility of each peak. And after that, we would get this loop plot. So the height of the loop here indicating how strong each enhancer is regulating each target gene. And of course, we want to select the significant loops. So we applied this algorithm to a huge pool of human cell lines, although it's a relatively homogeneous cell line, but we can still getting these peak-to-gene associations. And again, the height of the loops here indicating how strong each individual peaks is talking to the target gene here. And one thing I want to highlight is that if we downsample the number of cells that we use in this assay, or if we downsample the number of reads per cell, we actually dramatically lose the power to identify this peak-to-gene association. That actually highlights why we want to profile many, many cells in one assay, and also why we want to have high-quality cells. And then apply the same algorithm to the skin datasets. One thing we found is that, of course, we have many peak to gene associations with even higher fidelity, but also many of these peak to gene associations actually are not uniformly distributed around this gene, right? Actually, most of them are in the upstream of this specific skin-related gene. And accidentally actually found that many of this very dense region is actually overlapping with a known super enhancer region. So super enhancer is a concept proposed by Rick Young's group and also a few others groups roughly about 10 years ago. The idea is that, many enhancers are located closely in the genome, and they have very similar functions, and they kind of regulating targeted gene altogether. And because it's very close and very big, so that's why it's called super enhancer. And these super enhancers are known to be important to the cell identity and also to a lot of disease process, including tumor progression and others. And also, it's kind of hard to actually define these super enhancers because usually it requires performing ChIP-seq using histone modifications on roughly 1 million cells. And usually it's very hard to get this ChIP-seq experiment done in precious primary samples. So essentially, we find ways to use our single cell data to predict the location of the super enhancer without doing this kind of hard ChIP-seq experiment. And not only for this one gene, but for many, many of the genes in the genome, we found that their super enhancer gene shows many peak to gene associations, way more compared to the rest of the genes. So inspired by this, I'm wondering, is there anything special for these genes? Why do they have many, many peaks controlling them, right? So to further look into this, I simply rank the genes based on how many peaks are regulating them. So on this plot, you can see there are some top quiz that shows many peaks regulating one single gene. For example, the top quiz here is NOTCH1 gene, which has roughly 46 or 47 different enhancers regulating one gene. So there are massive regulating events happening for your one single gene. And also, if it goes down through this list, there are a lot of interesting genes like NOTCH1, LEF1, GATA, which are known to be important to the skin development. So essentially, we find the genes to narrow down, find ways to narrow down the genes that are important to the skin development. So to highlight our identification, we call these genes the DORC genes, and then these regions as DORCs, the domains of regulatory chromatin. We can also sum up all these DORC regions together as a DORC score, and we can see these DORC scores are very cell type specific. So each column here is a DORC, and each row is a cell type. So now we have the ability to directly link the chromatin readout with the gene expression readout at the same time. And then what happens to these two modalities if we think about more dynamic system? What is their temporal relationship, right? So to look into that, I look into the subset of the skin datasets. This is the TAC population, which is again known as a progenitor population, and it can differentiate to these downstream differentiated cells. So if we look at the gene expression in this differentiation, the 1-3 gene expression, it gets activated at a very late stage of this differentiation. And if we look at the chromatin signal, the 1-3 DORC, we can also see it at a very late stage, right? But if you look very carefully, we'll see these two activations are not happening at the same time, right? So the DORC actually activated a little bit earlier compared to the gene expression level. And if you subtract these two signals, you can define a residual signal here, which basically means for these positive cells, their 1-3 gene expression haven't changed yet, but their chromatin level already actually changed. So essentially, we think this is the strong evidence for lineage priming. So before the cells gene expression level change, the cells already figured out where I want to differentiate in the future. But why do we see this kind of lineage priming effect? So I further look into each individual peaks in this 1-3 DORC and compare that to the gene expression level. So along the differentiation, we can see the gene expression of either the intron or exon signal for the 1-3 again get activated at a very late stage. Before that, we saw a little bit promoter change in the chromatin level. seems to be happening earlier compared to gene expression. But way before that happens, we saw the activation of a few nearby enhancers. We thought it's probably the early enhancers get activated and then followed by a wave of secondary enhancers. Right? This is probably why we see this linear priming effect, because we have earlier onset of certain enhancers already. And to visualize this, I made a small cartoon here. So we are going to follow the trajectory from the TAC to the differentiated cells. And in the middle is the chromatin change at the same time. On the right is the gene expression level change. And so you can see here in this cartoon, even way before the gene expression level change, the chromatin level already have a lot of dynamic change going on. That's actually why we see the chromatin linear priming effect. So to summarize, basically we think we validated a very general gene activation model in the development, where we see the promoter, the enhancer get activated way earlier compared to promoter, and then we get the gene expression level activation. So now we know chromatin change roughly happened before the gene expression. Is it possible to use this information to further predict where the cell would like to go? So to do that, I developed another computational approach. We call it chromatin potential. Remember in shared-seq, we have the data in two different layers, and between these two different layers, the cells are always matched by experimental definition. So we're trying to find for each cells in its chromatin layers, what is their most similar cell in the RNA space? We think that's probably reflecting the cell's future state, right? And then we do this by looking at every single cells, and then because these two modalities are matched, so we simply find the cell back to its chromatin space and then draw an arrow between the two. We call it chromatin potential. So trying to predict the future of this current cell. So when we do this, we assume we know nothing about the cell trajectory and we know nothing about the time. We just want to do it blindly. Actually, surprisingly, when we do this very naive approach, we are able to draw a lot of arrows for the cells. You can see for most part, the arrows direction are very, very similar to the pseudotime directions that we defined in the pseudotime analysis, right? But also there are some differences, right? For example, here, we found two different routes which give rise to a few different lineages. And this expected route is the same route as we defined in the pseudotime analysis. But we also find a very kind of weird novel route here, which also seems to give rise to the medulla cells. And then this doesn't really support by the pseudotime analysis. And then we go back into literature and then think about actually how the cell differentiate in the skin. And what we found is that when we think about the hair follicle differentiation, we know the cells differentiate from the ORS and migrate downward, become the TAC, right? So that this TAC is a multipotent TAC. That means it can give rise to many, many different lineages. And then it further will differentiate into this uni-TACs. So it's a unipotent, only give rise to limited lineages. And then they can further differentiate to downstream populations. So essentially, the chromatin potential approach is trying to capture this heterogeneity within the TAC populations. We can also sort of validate this using other approach like RNA velocity. So, so I want to say that many, many times when we perform pseudotime analysis, we assume there is only one common route give rise to everything, but practically depends on the same system, depends on the time you are capturing all the cells, this assumption may not be valid. We really want to think about our assumption when we think about the stem cell populations. The pseudotime's assumption of a single route may not be valid, depending on the cases. So to summarize this first part of the talk, we developed a shared-seq that we can try to look into the similarity between the two types of definition. When we use cis regulation analysis, trying to capture the covariation between the two modalities, and we use DORC to highlight the key regions in the skin development, and eventually we use chromatin potential to predict where the cell would like to differentiate. So the shared-seq is great, but then what's the next? We don't want to stop here, right? So, so now after further optimizing the method, we are looking at a new version of the shared-seq. I won't tell you all the small details, but basically we optimized our TIM5 composition, as well as trying to incorporate poly-A tailing steps so that we can optimize both the chromatin and RNA recovery efficiency. If we compare the performance of the shared V2 to the commercial solution like the 10x multi-ohm, we can see our number of fragments in a TAC-seq or the number of UMIs in RC both outperformed or have very similar performance compared to the commercial solution, but we have much higher capacity. So because of the further decreasing of the sequencing cost, we thought we probably even want higher scalability in this kind of assay. So we expanded our barcoding space. Now, basically, we can profile about 20 million cells in one experiment, although the sequencing cost would be another inhibitory factor, but we can get there at least for the library preparation. And also with this huge amount of data, it's going to be a pain to really processing them. So we also optimized our computational pipeline so that we reduced our computational time. For example, even in the old pipeline, basically, there's no way we can run it through even within a month or one NovaSeq S4 round, but now we can process everything within a week. And then I apply the shared-seq V2 into the hematopoiesis system. So in the hematopoietic, we know we have the hematopoietic stem cells, which give rise to a few different lineages. So in one experiment, we profiled 7 different donors and roughly getting 900,000 single cell profile. So this is the UMAP from a TAC-seq. You can see these beautiful cell lineages. So the stem cell population is in the middle of this UMAP, which give rise to a few different lineages, including the B cell, monocytes, and also the infrared lineages. Now, with this massive amount of single cell TAC-seq data, I think we are at a better stage to really think about what we can do with this data. So one thing we thought we can do is to use that data to predict the binding of the transcription factors. So we know in the TAC-seq data where the DNA really cut into the genome, and this cutting could be blocked if there is a protein actually binding there, and this is called footprinting, right? And this protein could be a polymerase, could be a TF, or it could be a nucleosome. So in this section, I'm going to talk about how do we use the single cell TAC-seq data to predict the TF binding. So in the TAC-seq data, we transpose the DNA, supposedly have very uniform transposition, very uniform cutting. But in reality, actually, the cutting is blocked by this protein if there is a binding. So if we build a very simple model by comparing the cutting frequency underlying this protein binding region compared to the flanking region, we could calculate the probability, seeing what's the chance of there is a protein actually bind there, right? So we thought this could be a very simple way to predict the TF binding, but turns out it's actually very hard. There are 2 things it's hard. The first thing is, this is actually a real data when we look at the transportation events in the genome. And you can see it's actually very, very noisy. This is intrinsically because the TM5 also function as a TF. It's because it also have a motif binding preference sequence. And that's why even if you transpose into a naked DNA sequence, you still get very uneven cutting events. And when we use these uneven cutting events as a background information, we are kind of generating a lot of misunderstanding results. So we have to find a way to overcome this background information, right? So to do that, we prepared some artificial human genome sequence and then just transpose them this naked DNA and then see whatever data we would get. So again, you would get a very, very uneven transposition, even on the naked DNA, surprisingly uneven. And then what we did is we built a deep learning model trying to really predict how this bias would look like in the genome-wide. And then we can apply this model and then predict the bias. You can see this red track is the bias actually we predict. It looks very similar to the real sequencing data. And then we can, after we correcting this bias, we can predict the footprinting. So you can see this is the footprinting score. Basically, you don't really see any positive score because there is no protein actually binding the genome. But if we don't perform this bias correction, this is the result that we get, right? Because the intrinsic cutting bias would result in a lot of false positives. So we really have to perform the bias correction in a proper way. So after solving this first issue, then we actually have another issue here, is what would be the window that we should use to calculate this footprinting? Because there are, for different proteins that bind into the genome, they all have different sizes, right? Because for TF is small, it's roughly taking 10 to 20 different 20 base pairs as a size. But for nucleosome, they could be big. They could take roughly 150 base instead. Then what we should use to calculate this footprinting? We have a lot of discussion, and then eventually our approach is let's just do all of them. So so we did a screening of the window size. So from 1 at the bottom here, go up to 200 base at the window to calculate the footprinting. And you can see, like in this process, we have some signal, which is the prediction of where the TF actually might bind. You can see there are different patterns, right? This is a smaller protein, which we think is actually a binding of the CDCF. And then there are also larger ones, which is roughly about 150 base if you look at the x-axis. And we think these are probably the nucleosome binding. So essentially using our one model, we can predict the binding of both the smaller protein and also larger protein. And we can also validate our binding pattern by using ChIP-seq data. So you can see right in the middle here, we have a CDCF motif, and it's overlapping with a CDCF chip in the same system, as well as you can see this is the ChIP-seq data of crystal modifications. So right in the middle here, there is nothing, but on the side, there are actually crystal modification signal overlapping with the nucleosome binding. So actually the crystal modification happens on top of each nucleosome. So by combining this footprinting prediction with the motif information, we could actually very, very accurately predict where the TF actually bind in the genome. So we are validating our data comparing to the ChIP-seq ground truth. Although ChIP-seq is noisy, it may not be super trustworthy, but we still use that as the best ground truth for prediction. And you can see our predicted binding sites actually mostly overlapping with the ground truth with very, very few false positives. For the reference, if we only employing this TF motif as a prediction, we are probably have 40%-ish prediction power, but now we have much better accuracy. Now we have this working in a homogeneous population. We can also apply it to the hematopoiesis data sets that were generated, and we can track it along the differentiation or across different cell populations. So the color here is still indicating the probability of the TF binding that happens. So within this one single peak, which is only about 800 base, so we're really trying to zoom into one individual enhancer here. But even within this one enhancer, we could see their binding are not the same across different populations. We can simply cluster this data, and we can define several different clusters of enhancers. So essentially, even within a TAC-seq data, we saw one huge peak, but it could still compose of several distinct enhancers that may activate in different contexts. So we shouldn't really think about as one TAC-seq peak as one enhancers in many cases. So now we have the ability to predict the TF binding using a TAC-seq data. And then we're also wondering what happens between the RNA and also the chromatin level. So to answer this, we started a collaboration with Feng Zhang's group and the resident Julia in Feng's group. So we want to say, what if we perturb the system by overexpression the TF in the system? Does the cell's gene expression level going to change? Does the cell going to differentiate into different things? So we want to do this in a unbiased way. So essentially, we build a huge library and trying to perturb every single TF that we can find in our genome, roughly about 1,800 TF, but also included every isoforms of the TF. So in total, we did about 3,000 isoforms. And then we we do a very dilute infection, and then we let the cell try to differentiate for 1 week, and then we track how the cell differentiate. In this one experiment, again, we generated roughly 1.1 million single cells with more than 3,000 isoforms. And this is a huge UMAP gap. And as expected, probably most of the TFO expression doesn't really do anything to the human embryonic stem cells. But also surprisingly, we found that about 20% of the TF actually does have a function. In a traditional way, we would think maybe only a couple or maybe 4 or 5 TF may drive the human embryonic stem cell differentiation. But in our case, we found more than 20% of the TF actually have a function. So if we perform a stool-time analysis, we will say most of the cells seems to have not differentiated, but maybe a subset of cells here shows the differentiated cells. Then we can further cluster these cells. We can see these cells may seems to be directly differentiated into certain lineages, including different epithelial population, mast cells, or endocellular populations. So to further dissect what is causing these changes, so we did an MF-based approach, trying to cluster the different TF perturbation together. So you can see in this huge heat map here, we have a few different hotspots. Right? If we look at this first hotspot here, so each row here is a gene program, and then each column here is a perturbation of each TF. You can see certain TFs really work together to co-determine the cell fate decision. So this is the way we really want to use like an unbiased approach, trying to screening the function of the TF. But then we also want to validate our finding, right? So we narrow down to the neuronal differentiation in development in this case, and we found there are roughly 90 TFs may seem to have a function. So we just did another screening by pulling these 90 different TFs all together, and then let the cell differentiate. And then we use single-cell RNA-seq as well as imaging to validate our differentiation. So you can see after about 4 weeks differentiation, at the beginning, there is basically just the IP cells. There's no specific mark for different neuronal lineages or exercise. But after a few weeks, you saw a lot of red and green colors, and these are actually the expression of the neuronal related genes. So actually, our approach are able to really differentiate the cells into the neuronal lineages in an unbiased way. So hopefully I convinced you that we are able to really using our single cell approach, trying to asking what is the correlation between the TF binding, the chromatin openness, and also the gene expression level. And in the future, you can see that we can probably incorporate other modalities as well, and then really think about how these different modalities really work together as a whole to determine the cell fate. So with that, I want to thank everyone on this call, and also thank you very much for your attention. I want to highlight a few collaborators. Yan Hu in Jason's lab is leading the footprinting project. Che is our collaborator on the skin stuff. Feng Zhang and Julia for helping with the TF overexpression project. Tyler and Lindsay for the tumor progression project, as well as people in the SQL to help with the shared V2 project. Thank you very much. Oh, by the way, our lab is new, so we're definitely recruiting new people. So if you are interested, please reach out. Thank you.
Thank you so much, Sai. I know we have one question on the field.
Yeah, I can see.
I actually wrote down the question.
Oh, nice.
So it is, how were the data suitable for the footprinting analysis?
Yes, so the footprinting analysis does require many data. We cannot really do it on the single cell level because we want to build a model between the binding region versus the flanking region. And this, to make sure the model is accurate, we need roughly to aggregate about 500 cells. We could probably push it further to make it smaller, but 500 is the one we actually feel comfortable with. So basically aggregating 500 for these analyses. Yeah.
So I'm wondering about the NMF. Oh, so people can hear. I'm wondering about the NMF analysis that you showed towards the end. How did you combine, do a joint NMF factorization on this ATAC-seq and RNA and summarize the factors as is shown here in this heat map?
Oh, yeah, actually, in this case, it's only RNA. We didn't
Okay.
provide attack, yes.
That was my second question. What TFs would you get if you only use the RNA?
Actually, so yeah, so this is our RNA. We didn't do attack. But actually, in the second case, like in this case, when a more targeted readout, we did trying to perform attack as well. But because of the urgency for publication, we didn't do too much analysis on those. But I think because now we have the ability to predict the TF binding, and also we have the ability to perturb the cells. So I think the natural next step would be combining these two approaches, right? Say, what if we perturb them, and then what happens to binding, and then what happens to the downstream gene expression level change? I think you're probably asking what I want to do for the next year. Oh, there's another in the chat. Any pioneer factors footprints able to predict expression profile earlier in slow time? Yeah, I guess in this case, we haven't really looked too much in terms of the pioneer factor versus the secondary binding factors. But in general, we probably would expect pioneer factor binding first, and then you would expect like the binding of the secondary factors. But also in this case, if we're talking about the secondary binding, our footprinting approach is probably not the best model for this because they are not binding directly into the genome. We could probably like calculate the binding, but not based on the motif. So the prediction won't be very accurate. So probably a better way to do this analysis is not using the footprinting prediction. It's probably using either the protein level or the gene expression level as a better predictor. Do you consider the effect of cell cycle on chromatin accessibility? So in our skin data sets, we try to look into that, and we did find that the chromatin landscape actually is a very bad at reflecting the cell cycle information. Although we can see cell cycle and also the cell proliferation in the RNA level, but the chromatin level, we don't really see too much signal. So we kind of just ignoring that at this point. Let's see. Next question, is there methodological similarity between chromatin potential state techniques and the deconvolution techniques? I don't feel like there is any similarity actually in this case because our prediction is at single cell level, trying to look at the similarity between the two. And then for the deconvolution, it seems to be just trying to either deconvolute the either pseudobulk or trying to deconvolute the mixed population at per spatial spot. So I don't think there's much similarity in there. Is there any other question? Were there any completely novel cell types that were created in the TF overexpression experiments? I feel like that's a very hard statement to make, right? Because first of all, we are doing like very artificial thing. And also the cell states, I won't even call it cell type, are probably also artificial. And also the way we kind of classify these cell states are mostly by mapping to existing single cell data. That's already kind of biased our understanding. And even if we are able to find a combination to say it's a totally new, we probably don't know what they are at this point. And also not sure like whether it is actually meaningful in terms of like our at a clinical or biological setting. Yes.
So I have a question about the share seed. So for you looking at script, I don't know how much you work at cross cell types for those super
Mhm.
or is there evidence for any, let's say, partial species that might sometimes mutate to
Yeah, in the skin data, we couldn't find too many public available super enhancer data. Essentially, we only find two sets of data that is available. One is in the TAC population, the other is in the bulge, I remember correctly. And by looking at these two, we found they are both have a lot of overlapping with our prediction, with our dark prediction. I couldn't comment on, say, whether they are mutually inclusive or not. Maybe we need more super enhancer data to answer that question. And also because like for primary samples, it's very hard to get the super enhancer because of the cell number issue. So many people cannot really generate that kind of data. There's a new question here. So what are the barriers to systematically combinatorial overexpression TF with 4? I think for the experimental side, it's probably not too hard. We do have follow-up experiment trying to do combinatorial overexpression of TF like 2. But once you reach to more than 2, for computational side, this probably knows more, it's going to be very, very hard to tease out what's their actually effect in terms of determining cell fate. So we probably say we want to limit the number of perturbation per cell. And also if you're driving too much of many TF on one cell, then probably the cell fate will get messed up and the cell will probably die, I think.
I was interested in the calculation of the residuals that showed like the chromatin change in state before RNA. What was the math behind it? Is it related to RNA velocity or is it a different model?
Yeah. So that one is actually very simple. We simply we normalize both the chromatin readout and also RNA readout by looking at this by contact normalization, looking at 95 to 5% percentile to make sure all the signal are in the same range, and then simply subtract like minus. Yeah. So after so for that, also we first normalize all the data, and then we do a correlation. So for each of the chromatin profile for each cell, we calculate its correlation with the RNA cell. So the chromatin, we are looking at the dark signal. For the RNA, we're looking at the RNA expression signal. And we calculate the correlation between the two. If we don't know the direction, but we are calculating the correlation for each cell to all the cells. And then, if I remember correctly, I think we are choosing the top 10 cells and then smooth it as the future direction. So that means one cell is pointing towards the future, the average of 10 most correlated cells in the future. It's kind of a smoothing approach there.
So your main assumption is that ataxia happens before the changes in ataxia.
Right. Although when we do that correlation, we don't really use that assumption, right? Because just correlate. For defining direction, yes, we are using that, yes.
If not, let's thank the speaker again.
Thank you, everyone.